Good morning, and welcome to this session in our continued celebration of Barry Blumberg. This session reflects the intellectual richness of the society which Barry loved and enjoyed with such glee. It spans cross-species lung transplantation, politics, art, language, and mathematics. This morning's schedule will be slightly different from yesterday afternoon. We'll begin with a 40-minute lecture, then have a break, and then come back for two presentations. It's now a very great pleasure to introduce the Herman H. Goldstein Lecture. We are delighted to have with us several members of the Goldstein family, including Dr. Goldstein's widow, Ellen, and his daughter, Madeline. Welcome. After returning to UCLA and completing a degree in communications, Rothblatt went on to found several satellite communications companies, including Sirius Satellite Radio in 1990. Shortly thereafter, when her 6-year-old daughter was stricken with a rare and fatal disease, pulmonary arterial hypertension, Rothblatt reoriented her career. Pharmaceutical companies were not pursuing potential treatments very aggressively because the number of cases of this disease was small. Rothblatt developed a business model that would work despite the small number of cases and became an expert in a completely new field, earning a PhD in bioethics and building a highly successful biotech company, United Therapeutics. The drug this company developed has changed the face of pulmonary arterial hypertension. More physicians specialize in the disease, and its patients are living longer. Rothblatt is chairman and CEO, United Therapeutics. She holds a combined law and Master of Business Administration degree from UCLA and a PhD in medical ethics from the Royal London School of Medicine and Dentistry, Queen's Mary College, University of London. She is a member of the International Institute of Space Law, the International Academy of Astronautics, and the International Bar Association. Rothblatt was elected a member of the American Philosophical Society in 2008. Her presentation this morning will provide all of us with a glimpse into a different avenue of cure that she has been pursuing. It's entitled The Feasibility of a XenoLung by the End of the Decade.
Thank you, Barbara, for that very warm introduction. I appreciate it very much. I'd like to first pay homage to Herman Goldstein, whose contributions were essential to helping us win the Second World War and to the information society in which we thrive today. In this photo, he's second from your left, joined by Drs. Bigelow, Oppenheimer, and von Neumann, as well as the giant ENIAC computer to which he so essentially helped create. I'd also like to remember on this occasion Baruch Blumberg, with whom I had the great pleasure in January of a walk along the Atlantic coast near Cape Canaveral. XenoLungs are lungs from miniature pigs that have been genetically engineered to be tolerated when transplanted into humans. Let me start by answering why the focus on lungs and what is special about achieving this by the end of the decade. I'll then provide a definition of success and see how far we have to go to achieve it. Finally, I'll analyze whether it is possible to close this yawning gap in under 10 years. As Barbara noted, at age 6, our youngest daughter, named Genesis, was stricken with a rare lung disease called pulmonary arterial hypertension, or PAH for short. She was given less than half a decade to live without a lung transplant, as there were no medicines for the condition. Inspired by the film Lorenzo's Oil, and being myself a dyed-in-the-wool technologist, my response was to start a company to develop a cure. This company, United Therapeutics, is focused on the lung. Within 5 years, our company had created and achieved FDA approval of a new medicine for PAH called Remodulin. More approved medicines followed in the next 5 years. Thanks to these medicines, our daughter is doing well. But none of these medicines are in fact a cure. 3,000 Americans with PAH die every single year, and many more thousands overseas. This is why we remain focused on the lung. A lung transplant definitively cures PAH. No one has ever redeveloped PAH after a lung transplant. It is also curious that PAH patients survive worse than other lung transplant patients for the first year, but survive significantly longer thereafter. Chronic rejection or diseases associated with high levels of immunosuppression eventually undo the good of the transplant. The second lowest lines in this slide are the survival lines for lung transplants and heart-lung transplants. Only intestinal transplants fare as poorly. With XenoLungs, there is at least the opportunity to provide replacement lungs for rejected lungs, and more importantly, to use scheduled immunological pretreatment strategies to achieve a level of tolerance of the transplanted lung that is not practical with unpredictably available lungs from unscheduled human deaths. End-of-the-decade timelines have the ability to focus human attention on getting things done sooner rather than later. For example, in May 1961, President Kennedy said he believed that America should commit itself to achieving the goal, before the end of the decade, of landing a man on the moon and returning him safely to Earth. Prior to that month, all we had ever sent into space was a chimp. Most experts thought the goal was possible, but a huge stretch. As history turned out, absent that deadline, Apollo 11's July 1969 landing would surely have slipped to the 1970s or beyond. Similarly, in July 1990, I formed a company, now called SiriusXM, to create satellite radio by the end of that decade. During the 1990s, there were many land broadcasting interests trying to kill off the satellite radio idea. Their strongest argument was that it was premature for the government to authorize satellite radio because the technology was 20 or more years away. It was only because of tasking my company to the 10-year deadline that I was able to show the government enough technological progress to get the needed approval. Insistence upon the 10-year deadline got everyone motivated enough to figure out unusual ways to launch satellite radio right on schedule in the year 2000. Amazing things are more likely to be achieved and achieved sooner with such decade deadlines than without them. I think it is because within a decade separates science fact from science fiction. Hence, it is important for the in-a-decade goal to be viewed as doable. This talk will focus on whether XenoLungs within a decade are science fact or science fiction. Now, it is important to define what we mean by success. As this slide shows, the Salk polio vaccine was deemed successful in April 1955 with efficacy of 80 to 90% in a clinical trial. I believe our definition for success with XenoLungs is for survival to be as good as with allografts, meaning lungs donated from deceased people. The allograft survival rates for lungs are 92% at 3 months and 84% at 1 year. Hence, the challenge is, can we, before the decade is out, enable at least 84% of end-stage lung disease patients in a clinical trial to survive at least 1 year with a XenoLung? Let me now turn to the question of where do we stand at the beginning of this challenge. As this slide shows, pig organs with just one transplant genetic modification, called GAL-KO as a shorthand for galactosyltransferase knockout, have worked in primates for various periods of time. Clearly, though, the lung is the most short-lived. Indeed, I've been warned that the faster way to success with the XenoLung is to first achieve success with xeno-islets, then a xeno-heart, then a xeno-kidney, and then a xeno-liver. It is often hard to know in advance when the shortest path to a goal is a straight line or when it is not. In any event, let us continue to see whether just the XenoLung is feasible in a decade. Fortunately, we have a way to rapidly assess the likely tolerability of each kind of genetically modified porcine lung with a benchtop device at the University of Maryland Medical Center called the Isolated Lung Perfusion Machine. XenoLung candidates are explanted from genetically modified pigs and placed in a bath of fresh blood from human donors. This device provides these lungs with an opportunity to demonstrate gas exchange and interspecies compatibility. Functional parameters such as pulmonary vascular resistance are recorded until failure or elective termination at 4 hours. Thus far, we have tested XenoLungs with 2 genetic modifications, the GAL-KO I referred to previously, which prevents hyperacute rejection, and a new genetic modification called HCD46, which is a membrane-bound regulator of a part of the human immune system called complement. These lungs have also functioned for just a couple hours. Macroscopically, in this slide of XenoLungs in our benchtop machine, they actually look pretty good. But in fact, and this is at 3 hours, but in fact, they were very much clogged up with inflammation and coagulation and were unable to support life at 3 hours. This slide shows what happens when the same kind of genetically modified lungs with 2 genes have been transplanted into baboons, which are the most logical preclinical model for XenoLungs. By clamping off the native lung, we can see how long the XenoLung will actually support life. Baboons are separated from us and other great apes, such as chimpanzees, by about 30 million years of evolution, but they will still share with us key components of the immune and coagulation systems. Experts believe that if a XenoLung survives for several months in a baboon, it should also do likewise in a human. Thus far, XenoLungs with just 2 genetic modifications have supported life for under 2 hours in the baboon. But this is not a reason for despair. At the time President Kennedy challenged us to go to the moon before the end of the 1960s, the longest American manned mission in space, which had occurred just 2 weeks previously, was Shepard's suborbital Freedom 7 flight, and that was just for 15 minutes. Yet, by the end of the decade, we'd extended that 15 minutes to 10 days in our moonshots and to several months thereafter in the orbiting Skylab program. Hence, with the pressure of a deadline, it is possible within a few years to turn mere minutes across a seemingly deadly boundary into hours, days, weeks, and even months. How could we convert the deadly interspecies boundary into a safely transcendable border with XenoLungs? The answer to this question is twofold. Part 1 of the answer is we must use more than just 2 genetic modifications to bridge the 98 million years ago, or MYA, that separate us from the most recent common ancestor, or MRCA, with the pig. By the way, this is one of our very own 2-gene genetically engineered pigs, jauntily gracing the cover of Nature Biotechnology. Part 2 of the answer is that we must use the fascinating workings of the immune system as our friend to achieve a level of biochemical chimerism with the pig that recalls the macrochimerism inherent in the excellent size and function match between pig and human heart valves, hearts, and lungs. First, let's expand on the second part of this answer, embracing our immunological differences. I like to use this image of M.C. Escher's relativity to visualize how immunological chimerism, also called microchimerism, gets established. Our bodies are like a strange house with many hidden nooks and crannies, all eventually interconnected by surprising stairways. That some areas of the body had privilege from, or were basically invisible to, the immune system was recognized and exploited as early as 1967 by our society's president, Dr. Clyde Barker. If foreign antigen, such as on pig cells, can make it to these immunologically safe parts of the body and just occasionally use the lymphoid active stairways, then over a period of years, our bodies can become accustomed to them. We would then be microchimeric with such a pig genome. Such foreign antigen is, I think, much like a guest who has overstayed the 3 days for fish or friend rule. They seem noxious to us, but if our house is large enough, and if they can find a way to stay out of our way for a few years, they will probably have a house for life. More specifically, in this slide, you see in the upper left-hand corner that our immune system, shown as a skull and crossbones yellow diamond, makes an immediate determination when it encounters non-self antigen, such as that on a pig lung. The immune system divides all such antigen into two categories: deadly cytopathic threats and merely noxious, yucky threats. The deadly cytopathic threats send our immune system into a frenzy. It is willing to risk our own life to get rid of the cytopathic threat. Total war. And why not? Because the host would die anyway from things that look like the cytopathic invader. These kind of threats must be blocked at the border with genetic engineering and pre-treatment strategies, such as, but not limited to, the GAL-KO gene I mentioned earlier. We do not want our own immune system bombing us, tanking us, or calling in the Marines. However, if the immune system shunts downward to the yucky diamond, it realizes it faces a non-cytopathic threat. These are ones that our immune system knows are non-self and tries to eliminate, but relies on a much more limited repertoire of weapons to do so. Surviving cells from these more limited immunological attacks can hide in lymphoid-privileged parts of the body, as referred to in the previous slide, and are eventually tolerated, shown here with a peace sign. In other words, if the immune system can keep the leukocyte human Hatfields and the porcine McCoys from killing almost each last one of each other, the scientific term for this is reciprocal clonal deletion, then some of them can run and hide and have the pleasure of having Hatfield and McCoy children, grandchildren, and ever more distant descendants carrying on a low-level tit-for-tat fights for leukocyte generations to come. This pathway, which is the one that results in microchimerization, must be actively encouraged with both genetic engineering and pre-treatment strategies, such as, but not limited to, the CD46 complement regulatory gene I mentioned previously. This slide shows that the result of microchimerism is a biochemical self that is a lot more than the one genetically coded for in each one of our DNA. We start off life as the nicely formed capital I's shown in the middle of the picture. But as we proceed through life, the environment changes the complexion of our immune system, and we hybridize to the ever more strange-looking eyes around the periphery. To achieve success with XenoLungs, we want to actively encourage and shape this process of eye bending and eye warping by immunologically pre-treating the XenoLung recipients and the pig donors so that the recipients will be more receptive than usual to the pig's biochemistry. The trick is to not use so much pre-treatment immunosuppression as to put the donor or the recipient at risk of iatrogenic disease, including possibly graft versus host disease in the recipient, but also not to do so little as to make no difference. We want some funny pig eyes in our new immunological definition of eye. APS member Tom Starzl's early series of kidney allograft transplant patients were able to have the benefit of pre-treatment immunosuppression of both the donor and the recipient. These patients from the 1960s are now the world's longest surviving transplant recipients, have been documented to be microchimeric with their donors some 30 years later, many of them off all immunosuppression completely. However, as the transplantation field developed, such pre-treatment immunosuppression was restricted to bone marrow transplants because these were scheduled procedures. Pre-treatment was no longer practical for solid organ transplants because most of these became available as a result of highway accidents. A pharmaceutical-focused paradigm of very strong post-transplant immunosuppression has become dominant, with little tolerance for nurturing microchimerism via low-level immunological skirmishing. Xenotransplantation, amazingly, actually takes us back to the future. Because all XenoLungs would be planned transplants, there are a wide variety of immunosuppressive and chimerism-enhancing treatment regimes that can be applied to both the porcine donor and the human recipient before transplantation. This is indeed fortuitous because the vastly greater biochemical dissimilarity between pigs and humans would likely necessitate an intolerable load of post-transplant immunosuppression to just beat back rejection in the absence of microchimerism. Channeling the discoveries of Dr. Starzl over the decades since the 1960s, the current president of the International Xenotransplantation Association, heart-lung transplant surgeon Robin Pearson, has observed as follows. All available evidence suggests that induction of tolerance based on mixed hematopoietic chimerism should prevent adaptive immune injury to an allo or xenograft. With xenotransplantation, the genetically manipulated donor can be identified well before the transplant is planned and the recipient exposed to donor antigen under tolerogenic controlled circumstances, options not logistically feasible using organs from deceased human donors. Thus, Xeno offers a great opportunity to take advantage of a major advance in immunology to reproducibly promote antigen-specific tolerance. The next two slides provide a graphical image of the benefit of incorporating pre-treatment strategies into the xenotransplantation paradigm. Focus on the peak amplitude of the green immunosuppression curve along the top of the chart and the initial peak amplitude of the red immune response curve in the middle of the chart. Note that when there is pre-treatment, such as depletion of cytopathic bad guys that our immune system will never tolerate, the immune response, and hence the amount of needed suppression, is reduced. This is a good thing, as it reduces the risk of immunosuppression-triggered diseases that kill off so many patients. Here, in the next chart, you see that if in addition to depleting bad cells, we also infiltrate some yuck but tolerable cells, there is yet less immune reaction and the need for yet less immune suppression. While it is true that foreign leukocytes stow away on every transplanted organ, and thus every transplant patient is in some way microchimeric, it takes proactive immunological management to nurture a tolerable, but just so, level of microchimerism. This art is likely to be as important to XenoLung success as is selecting the right mix of genes to knock in and knock out. Now, I mentioned earlier that getting XenoLung survival from hours to years takes a two-pronged effort. The second prong, which we just covered, was to achieve microchimerism. The first part, to which we now turn, is to humanize pig lungs with more than 2 genetic modifications. This slide shows that there is an alphabet soup of over a dozen genetic targets that may need to be modified, including the 2 that have already been accomplished. Some of you may be familiar with several of these acronyms. For example, the VWF you see for von Willebrand factor in the coagulation circle. I want to emphasize that it may not be necessary to implement even half of the genetic modifications shown on the slide, or even all of them may not do the trick. Note also that some relate more to the immune system and some relate more to the coagulation system, but the 2 systems interact with each other. And also, there is a black box of other possible genes and systems of which we are as yet unaware. With xenotransplantation, we don't know what we don't know, and we always fear our success just leads to a new kind of barrier previously hidden from view by that last vanquished barrier. Hence, for many, xenotransplantation is but a Sisyphean task, depicted here by Titian. we must recognize that there may be systematic XenoLung incompatibilities beyond the fields of immunology and coagulation. Mammalian bodies are extraordinarily complex and intertwined. We will be lucky if after surmounting primary immunological and coagulation problems, we do not encounter novel showstoppers which themselves may trigger secondary immunological and coagulatory events. And we must also keep macrostructural incompatibilities in mind. Somewhere in that black box is the nasty fact that it will take a complicated surgery to do the right airway anastomosis in a double lung recipient, since pig bronchial anatomy has the right upper lobe coming off of the trachea. I think it is unlikely engineered pigs with fewer than half of the dozen genes shown in the previous slide will be tolerable, nor, as will be shown in a moment, will there be time during this decade to develop pigs with more than a dozen total modifications. Thus, we will have to be lucky, both with respect to pre-treatment strategies being very helpful and the black box being very empty if we are to succeed before the decade is out. So how are we going to go about tackling these dozen or so genes, pre-treatment strategies, and preparations for a human trial? My company, United Therapeutics, acquired the U.S. operations of PPL Therapeutics Company of Roslin, Scotland, the locale from which Dolly the sheep was cloned. As a result of scientific progress since Dolly's turn of the millennium cloning, we can routinely produce pigs with increasing numbers of genetic modifications. However, as shown in this slide, each genetic modification is a laborious exercise in transgene identification and insertion, shown here at the 1 o'clock position, followed by nuclear transfer and cloning, shown here at the 3 o'clock position, followed by gestation with hopeful viability, followed by careful assessment to ensure phenotypic expression of the desired results. Then there is a recloning of the best piglet expressor, shown at the 9 o'clock position, which will then produce a litter that can be used for our XenoLung experiments. It takes about 1 year to achieve testable lungs with any given genetic modification. Also, enough pigs must be produced at all times to enable us to study tolerability of XenoLungs with and without various different pre-treatment regimens. Because the number of possible genetic engineering permutations is so high, and because of the year time frame, it might seem hopeless that this quest could be accomplished by the end of this decade. Indeed, this slide shows that for the approximately 15 genes of interest, there are literally thousands of different genotypes possible if we limit ourselves to just a few genetic modifications that can be accomplished in a decade. But actually, things are not as grim as they may appear. We can use rationality and empirical data from researchers worldwide in the field of xenotransplantation to winnow down our choices. In order for us to succeed in this decade, we will have to operate in the lower left-hand corner of this graph to try about 100 different configurations of about 8 or so genes out of about a dozen genes of interest. It does help that we are also able to assess multiple genetic permutations in parallel. The process is somewhat like solving a Rubik's Cube. We need to work through the various combinations until we get it right. But what I do love about this metaphor is we all know that there is a solution to every Rubik's Cube. For example, let's see what good progress we are already making. As this slide shows, during the past 3 months, August, September, October, we created reclones, meaning pigs with confirmed high levels of desired gene expression for 3 different configurations of 3 modified genes, shown here with their gene names separated by periods. These reclone litters will be birthed over the next 3 months, December, January, February, and their lungs will be placed in the lung perfusion machine I showed earlier during the first quarter of 2012. This next slide shows that without even waiting for the outcomes of the lung tolerability studies with the different kinds of 3-gene engineered lungs, we will immediately use those genomes as part of the 1-year process of creating 4 different combinations of 4-gene engineered pigs. By mid-2012, we will have 4-gene engineered pigs with 3 different mixes of immunological genes and 2 different mixes of a coagulatory gene. For example, the recloned pigs born in July 2012, at the top of the chart, will have our standard GAL knockout and human CD46 genes I talked about previously, plus the gene for delay accelerating factor, also known as CD55, which helps modulate the immune system, and the important coagulation control gene called TBM for thrombomodulin. Moving right ahead, by late 2012, we will have several versions, just 2 are shown here, of pigs humanized with various mixes of 3 immunological and 2 coagulatory genes, 5 gene-engineered pigs, 1/3 of the way to addressing all 15 of the suspected genes of interest. All of this work is well underway and involves no breakthroughs. In summary, with a rolling production schedule of ever more modified pigs, we have the ability to create pigs with about 3 additional genetic modifications every 2 years. Let me emphasize again that many of these modifications will turn out to be dead ends, with worse results than lesser modifications. Other modifications may be eureka moments, and we can declare victory, perhaps because of synergistic effects with pretreatment regimens. We are even working on some technology to allow us to add multiple new gene candidates at a time, more than 1 gene at a time, and have them co-segregate upon breeding. Now, testing in machines and baboons will take an additional 2 years to complete from the date that each pig version becomes available. This slide shows that combining the pig engineering schedule I showed previously with a buffer of 2 years for testing in machines and baboons, we are looking at the mid-20-teens for a pig with 8 or so genetic modifications whose lungs might have achieved 1-year baboon survival and are ready for human use. But will the FDA or other regulators let us use these genetically engineered pig lungs in a human use trial? From the viewpoint of regulators such as the FDA, a XenoLung is a biological medicine, a sort you take just once for your whole life. Circled on the slide is a nutritional supplement for the lung that is certified vegetarian and kosher. I confess that pig lungs, we will never be able to achieve that standard. But I don't think the patients, Buddha, or Jehovah will mind. Seriously, the FDA must be persuaded that each XenoLung being produced is functionally identical to every other XenoLung produced and to the ones tested in the clinical trials, and that none of the XenoLungs are contaminated, adulterated, or infected with microorganisms, or even with strings of RNA that can produce disease in humans. While this is a tall challenge, it can be surmounted by building what is called a designated pathogen-free facility, or DPF for short, and operating such a DPF to the regulatory satisfaction in every way, from veterinary care of the pigs to the explantation of the XenoLungs, to their packaging and transport to the transplant surgeons at distant locations. The FDA has already laid out numerous guidelines to be taken into account for xenotransplantation, including archiving tissue samples for 50 years, and also achieving 1-year pivotal toxicology demonstrating efficacy without toxicity with special focus on monitoring immune-mediated toxicity. This picture shows the Philadelphia architecture firm Ewing Cole's conceptualization of a 1,000 pig per year solar-powered, biogas-heated DPF lung manufacturing facility. This DPF consists of a series of environmentally controlled and well-hydrated modules, each of which can handle about 80 pigs, and all of which are connected without exterior exposure to a central hub for surgical procedures. In practice, 80 breeding sows produce about 1,000 pigs per year. So one module would house the sows, while the other modules house the weaned DPF piglets with XenoLungs. A 3 times larger facility, if limited to PAH, pulmonary hypertension, would be needed to solve all the lung transplant needs of people with pulmonary hypertension who are now excluded from the transplant list. The Ewing Cole design here would accommodate 3,000 lung pairs per year and cover about 30 acres, including waste treatment areas. Such a facility would most likely be used to triple America's ability to provide lung transplants for people with end-stage lung disease, from pulmonary hypertension to pulmonary fibrosis, and from cystic fibrosis to emphysema and COPD. Architects have estimated that it will take about 2 years to build such a facility, and quality control experts require another 2 years to get it operational to FDA standards. Thus, the XenoLung DPF, designated pathogen-free facility, will have to start being constructed in 2013 in order to be ready to produce XenoLungs for human trials by 2017. Hence, only by building the DPF XenoLung manufacturing facility at risk, in other words, in parallel with determining the ideal genetic modifications and pretreatment protocols, will it be possible to complete a 1-year XenoLung trial before the end of this decade. The black rectangle shows the period of construction and process validation for the DPF facility, while the red dashed rectangle represents the period of populating the facility with the desired pigs to be used for ultimate commercial purposes, which for regulatory reasons must be the exact same genomic version of the pigs used in clinical trials. Lungs for transplantation should not be ischemic or without blood flow for more than 8 hours. This will require the rooftop of our DPF hub to be a heliport, and for us to deliver the lungs to transplantation hospitals that are not more than 6 hours flight time away. Most hospitals in the eastern U.S. are within reasonable round-trip helicopter distances from our intended DPF location in North Carolina. By combining a helicopter liftoff from our DPF with a charter jet flight from the Raleigh-Durham Airport, we can actually reach all major transplant centers in the continental United States. Hence, from a single XenoLung production facility in North Carolina, we could have the logistical capability to serve the entire American need for XenoLung transplants. This is a remarkably different paradigm than currently exists with the United Network for Organ Sharing for allotransplants. This new, almost FedEx-like logistical paradigm greatly facilitates the implementation of immunological pretreatment regimes to enhance organ tolerance. To satisfy the latent U.S. demand for about 15,000 lung transplants a year will require about 60 helicopter flights a weekday, or perhaps 5 per hour, 12 hours a day. That would be assuming that each helicopter took only one lung to one hospital. In practice, I'm sure we'll have some carpooling, or should I say, XenoLung pooling. Even with a suite of genetic XenoLung modifications that work for a year in a baboon, and with a designated pathogen-free facility that passes muster with the FDA, there is yet one further obstacle to meet our end-of-the-decade XenoLungs in man goal. This is the obstacle of clinical development. The clinical bar is to demonstrate better survival than with the allograft, which, as this earlier slide shows, is about 5 out of 6 survival at 1 year. A logical clinical trial protocol would be to enroll a few dozen patients with end-stage lung disease, but who are excluded from being listed on the current lung transplant list, and who have no other comorbidities that are inconsistent with at least 1 year survival. What kind of patients might these be? This slide shows the exclusions from even getting a lung allocation score, which gets you listed on the list. If we use these patients in our study, it does mean XenoLungs will start their competition against allograft survival rates with the additional disadvantage of a somewhat sicker population, but this is a common situation in drug development. On the brighter side, this disadvantage may help get the study enrolled quickly. There are many causes of end-stage lung failure, and about 9 out of 10 patients with these conditions never even get listed for transplantation. This slide shows the causes of end-stage lung failure for patients who are transplanted, and is somewhat similar for those who are not, with the exception that lung cancer would represent a large percentage as well. So in summary, to achieve an end-of-the-decade goal for transplanting a XenoLung in patient and returning them safely to health, it will first be necessary within the next 3 to 4 years to arrive at the right mix of genetic modifications and pretreatment strategies. Given the necessary timelines for cloning, recloning, and analysis, This means we must be lucky enough to achieve this with 8 or so genes, perhaps a dozen, but not more than that with current technologies. Then, to stay on track for the goal, during the middle 3 to 4 year period of this decade, roughly 2014 to 2017, 2 things must be accomplished. One needed achievement is to show that the chosen XenoLungs and pretreatment strategies reliably give us better than 84% survival in the baboon. This slide reminds us that baboons are separated from chimps and other great apes by 30 million years of evolution. They are readily available for conscientious use. The second needed achievement during the middle part of this decade is to show that we can reproducibly create and deliver those same XenoLungs to regulatory approval production standards in a DPF facility. Finally, in the last trimester of the decade, we must gain approval for, enroll, and carry out the clinical trial to show that our XenoLungs are as good as allografts. This slide reminds us that well-honed surgical techniques, such as those implemented at UPMC, are also key to producing dramatically better survival results. In this case, in fact, for lungs destined for patients with pulmonary arterial hypertension. It's these expert centers that have better than average survival rates where we want the XenoLung trial to occur. Now, beyond a doubt, converting the impermeable xenotransplantation boundary into a transgressible border before the end of the decade is a long shot. There are some 8,760 hours in a year, and as of now, the best anyone anywhere has done with the baboon is less than 8 hours. We are only 1/10 of 1% of where we want to be. But please remember that when the moonshot goal was set, we were also then only 1/10 of 1% of where we wanted to be. Alan Shepard traveled less than 200 miles high, and the moon is more than 200,000 miles away. And also, when I started my quest for Sirius satellite radio, we had 1,000 steps to go. Even without, we had no frequencies to use, there were no feasible digital coding algorithms, there were no flat patch antennas, and there were not powerful enough satellites, and there was vigorous politically ensconced opposition from the National Association of Land-Based Broadcasters. We began with about a 1 in 1,000 chance of succeeding. So this XenoLung in a decade challenge has its precedents. The keys for success will be: 1, take full advantage of the wonderful new tools of transgenetic modifications and cloning. 2, embrace, don't fear, some level of biochemical incompatibility via the nurturance of microchimerism with pretreatment strategies. And 3, don't confuse an unknown but definite number of challenges with a Sisyphean challenge. In other words, don't give up, but instead try harder and try smarter. I've not spoken much about money, but this project now costs us about $5 million per year, and the DPFs will cost about 10 times as much. Drug companies routinely spend a lot more than that to get a new drug approved. XenoLungs won't be any cheaper. But the benefits, if successful, could be much greater and more life-saving than any end-stage lung disease drug in development today. For this big bet to pay off for us in this decade, some shortcuts will be needed. We may not always have the luxury of testing every XenoLung element as a well-controlled experiment. For example, as mentioned earlier, we may need to add several genes at once, or we may need to test new genes and new pre-treatment protocols all at the same time. This all-up testing philosophy, championed by the main Apollo schedule driver, George Miller, shown here in headphones next to Wernher von Braun, was also essential to making the lunar landing possible in 1969. When President Kennedy leveled the moonshot challenge, all of the experts of the day were certain that the only way to do it was to build a nuclear-powered rocket called the Nova that had the power to lift off the Earth, land as a whole big rocket on the moon, and then blast off as a big rocket from the moon and go directly back to the Earth. It soon became clear, though, that this could not possibly be accomplished by 1969. Then a relatively low-level engineer named John Houbolt came up with an outside-of-the-box idea called lunar rendezvous. He said, why drag the whole rocket down to the lunar surface just to pull it all back up again? we can cut the payload mass in half by leaving the part of the rocket to get back to Earth in orbit around the moon, and instead just send the tiniest spacecraft possible for 2 astronauts down to the lunar surface, let them walk around, plant a flag, and then blast off just the top part of that tiny spacecraft back into a lunar orbit to rendezvous with the mothership. As noted in this slide, everyone who knew everything thought this idea was, dare I say, loony. Abe Silverstein, who named the lunar program Apollo and managed thousands of engineers, said it was absurdly risky to leave astronauts on the moon without the fuel to get home. Wernher von Braun, the guru of rockets, was openly disdainful and contemptuous. Yet in the end, John Hubolt's idea won the day, because while it did increase risk to the astronauts, it beyond a doubt cut in half the payload liftoff mass and thus reduced the size of the rocket to something that was possible with 1960s technology. It was, in the words of a famous NASA historian, a technological long shot. But the pressure of the end of the decade deadline and the inherent engineering bona fides of the lunar rendezvous to concept, plus John Hubolt's personal courage and persistence, won the day. Now we can think of no other way to land on the moon. We too will need some technological long shots to take us from a non-human primate. In the photo, it is America's first chimp in space, Ham, to a successful XenoLung transplant in a decade. I believe there is a way to achieve this goal using outside-of-the-box thinking, rearranging the pieces of the puzzle, and pairing scientific brilliance with cogent persuasion to cut a factor of 2 or more out of the challenge. In short, I am looking out for the John Hubolts of XenoLung. It seems to me that the project is science fact rather than science fiction, and hence an end-of-the-decade deadline may be just what is needed to help us achieve success. Thank you.
Fabulous. You'll take some questions. You're so good. So we have time for several questions. Please wait for a microphone and remember to introduce yourself before you ask your question.
I'm Maynard Olson from the University of Washington. Well, thank you for an inspiring talk. I'd like to ask a question about the culture of innovation. I'm a basic scientist, and in general, we thrive on uncertainty and unknowns, but we don't like timelines. Whereas my engineering colleagues tend to thrive on timelines, but they don't like uncertainty and unknowns. So I'm just curious, in your experience, is it the basic scientists or the biotechnologists that are the hardest to infect with your passion and enthusiasm?
I find both. Thank you for the question. And I had the great honor early in my career to be mentored by Sir John Fane, who was responsible for discovering the mechanism of action of the molecule that we use to treat pulmonary hypertension today. John was a champion of basic science, and I am the champion of basic science. And I think that the beauty of basic research is that it's kind of the flip side of you don't know what you don't know. And surprising things come out of basic research that could leap over many careful steps of engineering and project development. So I find that both the engineers and the basic scientists are very excited. I know in our own project, we have basic scientists working on the xenology, the basic understanding of microchimerism. They're frankly excited that somebody is pouring money into this field. And the engineers are excited that they have a concrete project to work on. So I think both can get infected with enthusiasm.
I want to thank you for an excellent talk. Paul Marks, New York City. I was wondering, this may be very simplistic, would attempting to partially remove and transplant the lung, one of four lobes, be an avenue that might be quicker, easier, and perhaps safer in terms of your long-term development plans?
I think it's actually not simplistic at all. It's a brilliant question. One of the real breakthroughs in lung transplantation has involved what's called living lobar transplant, in which one relative donates one or two lobes, I think generally two lobes of a lung, ordinarily to a child suffering from a end-stage lung disease. So that certainly is a possible avenue. It works, and those patients do pretty well. With regard to the testing in the xenotransplantation area, all bets are open, and that's a good idea, and I'm going to go back and talk to other members of the team about it. I think it's really important to be like John Hubel. The most important ingredient to our success is to be open-minded.
Chris Russell from Harvard. I'm just would like to hear you reflect. It was a fascinating presentation. Using this man on the moon analogy, that has been used over the last 40 years frequently, but it's a technological challenge. It was used in the launching of the war on cancer, which was more of a biological challenge. So what do you see? It's a great analogy to set the deadline, but what do you see as the biological complexity and the difference? This isn't really sending a man to the moon. This is a different kind of challenge. And so what do you see as the difference when you're thinking of that analogy? What is greater in your challenge, or do you see them as analogous? Obviously you do, but what's the complexity on the biological addition?
I'm so glad you asked that question because I had a really nice 10 minutes or so comparing and contrasting the utility of that analogy that to get this under 45 minutes I had to cut out. So it's an excellent question. I've thought a lot about it. There are some excellent histories written of not just the whole Apollo moon program, but even the decision that led Kennedy to announce this quest. Frankly, I think that the analogy is not the most accurate. It might be a little bit more accurate to say, what if the challenge had been to go to Mars and back in a decade? Because if the goal is to survive 10 days beyond a generally impermeable boundary, that's what we achieved with Apollo. And we were able to basically get astronauts to live in space for about 2 weeks. If the goal had been to have them live for a year, we could not have done that in the 1960s. We weren't there yet. We did get there in the following decade, and the Russians got there for sure in the following decade. But it's a great question because I get pressure on me, and I promise you I'm going to get a whole lot more pressure on me that says basically, Martine, you've set the bar too high. Why a year? Why not 6 months? Why not 3 months? Why not 2 months? Why not 1 month? Maybe if you do it 1 month, the person will get an allo transplant within a month, and then they could, go on to have that survival curve that you saw for the allo transplants. But I've been educated well by the heart and lung surgeons, and we, of course, in this audience have some of the greatest and among members of our society. A lung transplant operation is a horribly invasive procedure that goes without saying. And it's not something that you should undertake flippantly, and I don't really feel that it is appropriate to undertake it unless you can give somebody an expectation of at least a year, a reasonable expectation of at least a year of life. Of course, there's no magic numbers. A year is just a round number, but I would use that as a rough benchmark of what's reasonable.
It's a question up in the balcony.
Yes, this small, Wendy Lamb, New York. The small size of a miniature pig's lung, what kind of capacity would that give a human recipient?
Great question. Same as the human lung. The miniature swine is not really. It's only miniature compared to the very large pigs that one's used to see used for agricultural purposes. Somewhat tongue-in-cheek, some people call the miniature swine pigs pygmies, but they actually are as an excellent size match for the human heart and the human lung, and we would have the same vital capacity. They have a natural life of around 25 years, so there would be a hope that each transplant could hopefully last a long time.
Erwin Shapiro, Harvard. Trying to think a little out of the box. I was wondering if it's totally crazy to think of having not so much a lung replacement, but a lung addition. Could the obvious problems be surmounted to supplement rather than replace?
It's another brilliant question, Dr. Shapiro. As many of you may know, there are instances in which hearts are transplanted into people while keeping their native heart. And ultimately, actually, the transplant can be removed because the native heart has recovered over time. I'm sure that will be explored. It could be a little bit easier path to FDA approvable clinical trial, because if something does go horribly wrong between the baboon study and the human, you can at least take out the baboon, the XenoLung, and the individual would not be much worse off than they were before. Of course, the surgery is traumatic, but I've gotten already 2 great ideas from this lecture, so thank you so much for the opportunity.
Howard Gardner from Cambridge. This in a sense does follow up from Paul Marks and Erwin Shapiro's question. You gave a very rational account of how you're proceeding, and it was impressive, but you also used the word luck a lot. And so I'm interested in the role of intuition, and particularly in the end you have to decide which things to support and which not. And if somebody has what they said with reference to Barbara McClintock, a feeling for the organism, and says, look, of these 20 different things, my intuition is it really should go that way. How do you deal with that? And where do you make those bets?
It's tough because I think it would probably be obvious to everybody here that in a group of, say, 10 scientists, people might have 15 different preferences for which of those genes to go first with. And different scientists are working on different genes. What I'm trying to do is to pursue a path of diversity by having different research groups pursuing different combinations of genes. Ultimately, there is going to be a need to make this kind of gut feel, this intuitive judgment. We're trying to hire more experts that could advise on that. But ultimately, the main skill set, I guess, that people expect of me as the head of the organization is to actually just make a decision and be willing to live with the consequences that it might have been the wrong call.